Genome evolution by recombination hotspots. Recombination hotspots are the regions of the genome that exhibit elevated rate of the recombinations when we compare it with other regions of the genomes. So these are actually those regions on the genome where the recombination rates are increased as compared to other regions of the genome. Recombination hotspots are thought to arise due to higher order chromatin folding in the chromosomes that becomes accessible for the recombination. The peak recombination rate within hotspot can be hundreds of times greater than those regions where we do not see the rise of the recombination rates. Recombination hotspot can occur through meiotic recombination as well as through errors in DNA duplication. So there can be two different reasons of recombination hotspots. One of the reasons can be meiotic recombination, and the second reason can be errors during the DNA replication. Linkage disequilibrium has identified more than 30,000 hotspot regions in the human genome. In humans, average number of crossover recombination event per hotspot is approximately 1 out of 1300 meiosis. And in certain areas of the genome, it has been observed that the frequency of hotspot recombinations is 1 out of 110 meiosis. So, in certain areas of the genome, it has been observed that the hotspot per crossover can be 1 out of 1300 meiosis, and in certain areas of the genome, it can be 1 out of 110 meiosis. Just to understand that what is recombination hotspot in case of gamma globin genes, here we have seen that there are TG repeats here in the middle of the exon. Just take this diagram as an exon. In the middle of the exon, there are TG repeats. So these TG repeats, they actually function as, or they work as hotspot regions for the duplication of the genomes. Recombination hotspots do not seem to be the solely caused by DNA sequence arrangements or the chromosome structure. Means it is not just due to the sequence of the DNA, or it is due to the structure of the chromosomes. There can be alternative methods by which there can be recombination hotspots can occur. Alternatively, initiation sites of the recombination hotspot can be coded for in the genomes. Recombination hotspot and current isochore structure. Perhaps difference in the recombination rates have caused the current isochore structure of the genomes that can be due to the biased gene conversions. So if we conclude that what is recombination hotspots? Recombination hotspots are those regions of the genome that exhibit increased rate of recombinations. These regions, they are source of genome evolution. So the difference that what is a normal recombination rate and the increased rate is that these are those regions on the genome where there is increased rate of recombinations when we compare it with other regions of the genome. So those regions, they are called as recombination hotspot regions, and they are also responsible and are considered as source of genome evolution.